Okay, so I had a request to talk a little bit about the class project in case you guys are already starting to think about what you want to do for your final project. It's a big chunk of your grade, so it's good to start early and know what you want to do and do a good job. This is also your opportunity to do your research and also get class credit for it. Yay, right? Double dipping. So, and you can come into class for, we have like 4 class periods set aside to work on your project. So you can basically come to class and get help from me and Narula to do your research, which seems like a pretty good deal. But it requires, knowing what you're going to do and planning well and utilizing your class time. I will tell you, like 2/3 of the students usually don't show up to those class periods. But the third of the students who do are usually the ones who do really well and get a lot done by the end of the semester. So showing up to those class periods and starting early is definitely going to be an advantage. So this is what the syllabus says. You need to identify a bioinformatics research goal and an appropriate data set, execute the project, and prepare a written report with supporting data, analysis methods, code, and other documentation. I'm only going to ask for a 1-page report as far as like describing your project. What I really want to see is that you're able to do some informatics analysis. And if you pick something outside the scope of what we've covered in this course, that's fine with me. I'm maybe less helpful. Like I've had people pick proteomics projects in the past, and that's fine, and I just don't know that much about proteomics, so I'll do my best to help you. But if you pick something like variant calling or RNA-seq, I'm probably going to be more helpful. But again, this is your opportunity to sort through some informatic challenges for your data of interest. So you can pick something outside the scope of what we're doing. It does need to be informatics and not like, I'm going to extract DNA. That's not going to fly. We'll do oral presentations. They might have to be a little bit less than 15 minutes because we have a lot of people. We'll go through, I'll create an actual like grading rubric so you know what your goals are, but I haven't done that yet. But since I had already had the email question and I wanted to give everybody the same advice, and it's in the syllabus, this how to be successful in this class. The last one is probably one of the most important. Select a final project that is a proper scope to accomplish in 4 weeks. If you go too big, you're gonna get frustrated and you're not gonna get anything done. If you go too small, I'm gonna be underwhelmed and not likely to give you a very good grade. If you're like, oh, look, I took some data and I trimmed it. I'm like, no, that's not enough. So the proper scope is really essential. Most students tend to go through for too big. So try to find something bite-sized that you can do, run a few pieces of software, get to some intermediate result. This is not necessarily like, I'm gonna write a whole chapter of my dissertation and have a publishable unit.
Can we get feedback from you on the scope of the project before starting it?
Yes, absolutely. I think this is all, hold on. There's a 1-page project proposal that is 10 points of the final grade. And hopefully that is annotated, noted somewhere on some day at some point. Oh yeah, the schedule's at the bottom. Good job. I don't know how well we're sticking to these homework dates either. I think we might actually be on schedule with those this semester, which is good. Oh, project proposal, November 5th. That is way far away. Start earlier than that. I probably should have made it due earlier than that. But you'll send me a project proposal and I will tell you, I think this is crazy, don't do this, or this is great, add something else, or this is wonderful, continue on. If you give me enough information. Are there any questions about that? And if you want to ask me ahead of time and just like be like, hey, I was thinking about this, feel free to email me or catch me after class. I'll give you feedback now. Any other questions? Okay, so there's one of the steps for the lab is going to take a really long time. So I'm going to do like half the lecture, and then we're going to go do some lab, and then while that like one thing is running, we're going to go back and do some more lecture. So we're going to start talking about variants today. So variant calling, with short reads, the most common things that we are looking for are SNPs, which are single nucleotide polymorphisms. I also see in the literature a lot SNVs for single nucleotide variant. I don't know why we need both, but we have both. Indel for insertion or deletion. Most of the times when I see indel, I think they're referring to short, like 1 to 10 base pair insertions deletions. But I think you could technically use it to talk about large insertions and deletions as well. But usually with short reads, you're constrained to detecting relatively short insertion deletions, at least within the scope of a single read. There's a whole different suite of software you can use to find really big insertions and deletions. But the basic strategy here is to examine your read alignments and look for differences between the reference and the individual being sequenced. So this depends a lot on what the reference is and how different it is from the individual or individuals you're interested in. There are a number of difficulties that make this process harder. Cloning artifacts are a big one. So if you introduce an error during PCR and then continue to PCR, you just amplify that error, and then that looks like a real thing. So that's a problem. Errors in the sequencing reads specifically from the instrument, also a problem. The instrument isn't able to call a base very well. Incorrect mapping, so if your mapping reads to the wrong location in the genome, you'll potentially think that there are variants in that region that aren't there. And this is compounded by errors in the reference genome itself. So if your reference genome has been misassembled or has an error, which totally happens, you may actually have the correct variant or the correct nucleotide in your data set, and the reference is the one that's actually wrong and there isn't a real variant. Those are hard to detect. So Hingley, the developer of BWA, all around generally just an amazing bioinformaticist, wrote this paper looking at major sources of errors and variant calls. And he concluded that the two biggest problems are erroneous alignment in low complexity regions. So low complexity region might be where you have a whole bunch of T's, or you have AT, 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 AT. And it makes it very hard to align because you just don't have enough information. Anything that's repetitive or just generally doesn't have a nice random mixture of A's, T's, and G's can be a problem. And number two, the incomplete reference genome with respect to the sample. So if your reference genome is wrong or incomplete and your mapping reads back to it, it's really hard to discover variation because you don't know what the base is. You don't have a solid foundation on what you're building. So both of those things we're going to talk a little bit about some advances today that have come along to help combat that. This is an example of a misalignment in a low complexity region. So if we have this reference sequence, and these stars have just been put here for the purposes of the alignment. So the reference sequence actually it's just these nucleotides. We have a read that perfectly supports the reference. It's going to get aligned totally correctly. We have a read that supports the insertion, and because we have a full set of bases on the left and a full set of bases on the right, we can see that there is an insertion of a GC. So maybe this is a diploid and it's heterozygous for this little insertion. But if you have this read and it ended right in the middle of the insertion, you might try to call those SNPs because you can't tell if it's an insertion or if those bases have changed. Similarly with this read, if that insertion is toward the end, you might try to call these SNPs. And you wouldn't know that there was an insertion because you didn't have enough bases on the other side to confirm that there was a real insertion. And so when the alignment program, when the variant caller tries to go in and look at this, it says, oh, I don't see that much evidence for this insertion. I don't see that much evidence for these SNPs. I don't see that much evidence for these SNPs. This whole thing's just a mess and you don't get any variance. So you have to go in and do a lot of extra work to figure out that this is an insertion and that all three of these reads actually support the same insertion. This is from Hingley's paper. I'm not going to go through it because I feel like I have to stare at it for 10 minutes just to explain it to myself, much less to explain it to you guys. But this one is based more on the problem of having like these small 10 to 15 nucleotide repetitive sequences where you just continue. It's very much similar in the same vein as the previous example, just a little more complicated, where there's an insertion of a GGGAC and it gets mismapped because it's the GGGAC is inserted and it matches the exact next 6 nucleotides. It's just very hard to sort out that you have a big deletion here. Take home message. Indels, far more problematic to deal with than SNPs. If you have a SNP, and whatever's on the right side and whatever's on the left side are identical, you're probably going to call it correctly. When you have insertions and deletions, your alignments get all wacky, and it's very much more difficult to figure out. So we're going to be focusing on using the Genome Analysis Toolkit. There are a whole bunch of other programs that you can use for variant calling. This is the one that I'm the most familiar with, so we're going to use it as our example, but I'll give you a list of other ones. It's open source, and it was originally published in 2010. But the great thing about this program is they really continue to expand and improve every year. They have gotten lots of support from the community to really, I mean, they rewrite their underlying algorithm to make it better all the time. It is a complicated pipeline. We will only see a little tiny piece of it, but it's worthwhile. I will say that it's a very, it's a robust way to call variants. In January of this year, they came out with GATK4. Their big new thing is that it runs on the cloud. So you can take these fancy workflows and load them up on Amazon, or they have something called Fire Cloud that they run at Cornell, and you can just pay to shove all your data into it, and it all runs, and it comes back. And if you're interested in doing that, they'll give you $300 in cloud credits for free to just get started. So if you're thinking about calling variants, I'd recommend you try that out. We haven't tried it yet, and I'm really excited to try it. There's a whole bunch of new features. It covers SNPs, indels. They have recently added copy number and structural variation, which I haven't even tested yet. They also do both germline and somatic mutations. So somatic being like only a few cells in your sample maybe have the variant of interest. And so you're not looking at nice ratios of 1-to-1 or the entire sample shares the variant. You're looking at maybe 10% of the DNA copies have that variant, or 2%. And with enough depth, they claim that they can robustly detect somatic mutations, which I'm sure there are other good use cases, but cancer is the one that has kind of driven a lot of the research in this area. And it is for both genomes and targeted sequencing assays. So if you're doing whole genome resequencing, great. If you're doing GBS or RADseq or exome capture, it will also work for those. I would recommend you go in and look at their user guide. I like the top, it says very complete, much documents, so quality, wow. They have the GATK people have a sense of humor, which is great. And so there's a deep dive here. Like there's hundreds, maybe thousands of pages of information once you get in here. So I'll point you to that resource. And this is the GATK on Fire Cloud, which should be simplifying all of their workflows. So we're gonna run through this real quick, and I'm gonna break it down. But this is their vanilla pipeline for germline short variant discovery. So this is something where you expect all of the samples, all of the cells in your sample share the variants you're gonna call, and you're only looking for SNPs and indels. And so it's kind of the most vanilla of their workflows. The first thing you do is data pre-processing. And we've already seen a little bit of this in the sense that we've gotten to the point of trimming and mapping to the reference. They do list BWA as still one of their preferred mappers, so that's great. The first thing when you get into the GATK pipeline that they want you to do is mark duplicates. So that's where if you did have some kind of PCR artifact, the read is going to likely have the exact same start and the exact same end base, and they're gonna be 99.999% similar. And by having that starting base and ending base be the same, they can say, I think this is a PCR duplicate. I'm only gonna keep one copy. And then they have this recalibrate base quality scores. And basically, these authors have said, we don't think that the quality values that come off the instrument are good enough. We're gonna do a whole bunch of fancy statistics and renormalize all those quality values and make them better, make them more accurate to reality. And they may have a point. The instrument makers are, it behooves them to make the quality look as good as possible. Whereas if you're calling variants, you really want to have anchoring in reality for quality values. So they claim that this really helps with being able to detect variants accurately. At that point, you have a new BAM file. The second stage is actually to call the variants. They use something called the haplotype caller. This is a super smart variant caller, and basically what it does is it throws away all the assemblies or all the alignments that you already did. It says, okay, your alignments, those are cute. Like, I can see enough variation to think there's probably a variant here. But I don't believe, like, all your alignments probably suck. So I'm going to throw them all away and start over. And it does actual assemblies, brand new haplotype assemblies for the region. So it tries to come up with, you tell it your diploidy of your organism, haploid, diploid, tetraploid, whatever. And it tries to come up with the actual real reference to 1, 2, or 4 copies of the reference genome in that area, and then remap the reads back to those references. No, you still have to do the mapping step because it doesn't want to do this for the whole genome. It only wants to do it in regions where there's variation. So you could go, 100,000 bases with no variance, depending on how similar your samples are. And it's only going to go right back into where there's a variant and do the local assembly because it's very computationally intensive. If you tried to reassemble the whole genome, it'd take too long. So they only want to do this in those little localized regions. So it does use your original read mapping. It's kind of like, let me go back. When you have this, and it says, well, none of that really makes sense, but I see lots of little things, like lots of things look like they're different. So I've got maybe a 10 base window in which I think there's variation, but I think these alignments are not good enough. And so it would actually like get rid of the reference, reassemble these reads, figure out that GC is an insertion, and then go back and map it back to what they think is the new reference instead of the old reference. So it's a lot more accurate, if that makes sense. Yeah, so that's kind of how it works. It does a really good job. And the output format, yes, we get to learn a new format today. BCFs and GBCFs is variant call format. So that's what you get your output in. And then they have another step where you merge, if you have multiple samples, you merge them, and it tries to use the information from all the different samples to make a better variant calling. So if you had, let's say you had enough depth in one sample to confirm there is an actual variant at this locus, you can go back into another sample where you had less depth and maybe be more convinced that, we know that variant is real from individual A. So if we see that base in individual B, we're going to assume that it's probably real there too. Does that make sense? Like if you pool all the variants from your entire population and you know where they occur, then you can go back into each individual and be more accurate. Oh. So what you can do is you can call variants using one type of data, and then you create a database and you feed the location of those variants back in as priors. And it will use those priors to call your new variants. PacBio is like 15% error, so all of the variant calling with PacBio is totally different than the error. The Illumina is 99% accurate. PacBio is like 80 to 85% accurate. So variant calling is a whole different ballgame in these two types of data. You can always call variants in one and then use those as priors to call variants in the other. Is it like you could get a database of like, I know these 50,000 SNPs are legitimate, they exist in this population. And you can feed that back into the software and say, please go look for these. I know they exist. Does that help at all? That's how I would do it. I wouldn't try to I wouldn't try to do one analysis with two types of data necessarily. Okay, and then the final step with variants is always filtering, because sometimes you get things that don't make a lot of sense, and your stringency is very much dependent on what you're trying to do with your data. If you really, really only need things that are extremely high quality versus if you really want maybe things that are less high quality but contribute to whatever your biological question is. So they have this variant recalibrator. They use machine learning to identify variants that are likely to be real, and then assign this more reliable quality score. Sure. So GVCF is a tab delimited format, and every sample, it just adds more columns. So if you have 100 samples, you have 100 extra columns, and it gives you the genotype in each and every sample all the way down. So it's technically infinitely expandable as long as your computer will let the lines be infinitely long. So a GVCF is a constraint on a VCF that I meant to add a slide on this and I didn't do it last night and now I can't remember. We can look it up. I forget. A GVCF is a proper VCF. It meets all of the specifications of a VCF. It has additional specifications on top of it that at the moment I can't remember what those are. We can look it up. Okay. So this variant recalibrator is awesome if you work in mice or humans or Arabidopsis. But if you work on some brand new system with no prior data, you can't really use it. It requires a lot of data, so it requires a lot of depth per sample, and it requires a database of known variants to do the machine learning because it has to do a training set. So it uses the known variants as the training set. So if you don't have a database of known variants, you don't have sufficient depth, this won't run. There are other ways to filter. We'll see VCF tools filtering during the lab, which is a much more simplistic way of filtering your variants. Bode Olukolu in my department is like our genotyping guru. He's going to come do a 2-day session with you guys in November and have a whole bunch more to say about this. Yeah.
Okay, this is a super basic question. What is filtering variants?
Oh, just saying, this has a depth of 4, and I have 3 A's and a C, and I have no prior evidence that the C exists, and we're calling it a heterozygote, but I just don't feel good about it, and I'm going to throw it away. Just giving some kind of threshold of belief is just quality control. Is you get to go in and say, I want to see at least 10 reads covering this region for me to believe that I've called the genotype correctly. Or I want to see the map quality. I only want to use reads of map quality of at least 10 to use to put into the variant calling. Or maybe you're actually, another type of filtering you might want to do is that if your depth is way, way, way too deep, if you did 10x coverage of your whole genome and you have a region with 500x coverage, that's a repetitive element. You want to toss all of that. So there's like all these different ways you might just want to get rid of things. You're just getting rid of things you don't believe. And there's lots of strategies for that. The machine learning pipeline is nice, like I said, but it requires a lot of prior.
So you don't believe, or that you don't believe, and or they just don't, they don't address your question. So you just.
That too, yeah.
Okay.
So things that like they're on the border of statistical significance, but maybe they're just not good enough. Like if you're going in to do a genetic linkage map, you really only want things that you have called correctly in every individual, or you'll get distortions in your linkage map. So you may have a super, super, super high filtering level. But if you're doing more like population structure, and you figure as long as 90% of them right, my population structure will come out in the wash correctly, maybe you don't filter stringently. Oh, and I was looking at papers last night when I was preparing this lecture, and I found one specifically for ancient DNA variant calling. So if you're interested in this, I'll send it to you. Because I was thinking about you, because you would be like a really good example. It might behave kind of like somatic variation, where you'd only have like some cells. If they were degraded, you'd actually want to filter those, but you might see like variants that like occurred. Anyway, we can talk about that after class.
Right, with mitochondrial DNA, maybe like there's going to be more mutations in mitochondrial muscles and other parts of the body.
Yes. Yeah. So all of this is very dependent on like what questions you're asking, what samples you have. I think it's hard to teach this class in a way that I think it's actually going to help that many people, because so much of it's so variable and dependent on what you're doing. But if you learn enough command line, then you can do whatever you need to do. That's my philosophy. So for the lab, we're going to use GATK3. This is still an OK pipeline, but I'm going to recommend you guys upgrade to GATK4. I kind of messed up because I thought GATK4 was a pretty small upgrade to GATK3, and I tried to run it over the weekend, and I realized it's totally different. Like it's 100% different. Like I need to spend a whole week relearning the package, and I didn't have a whole week. So we're gonna do GATK3 because I know how it works. But you guys should totally learn GATK4. I think it's basically, I got tripped up immediately because I've always done this indel realignment, which is where there's like variations around insertions and deletions are messy, and you have to go in and you have to fix them. And basically what they have done is they have moved that step out of an independent command line call and into their haplotype caller package, and it's kind of done automatically. but I couldn't figure out how to make it run correctly. So we're going to go back and do the GATK3, which is how we called variance for many years prior to GATK4, and it worked fine. But you guys should definitely, if you're going to do this, go put the time in, go get your $300 worth of FireCloud credits and figure out the newest version. One of the ways, if you don't have GATK4 running and getting their like super fancy new high quality values, the original kind of statistical cutoffs, or the way we do stats for variance is genotype likelihoods. And that's going to calculate the probability of the observed data given each genotype. So it'll say, like, let's say we have a diploid organism, and we can either have homozygous for the reference, heterozygous, or homozygous for the alternate. It'll say, in case A, homozygous reference, the likelihood is very low. Heterozygous, the likelihood is very high. And homozygous for the alternate, the likelihood. So like it should give you a distribution of like what's the likelihood of each of these things given the data you showed me. These are usually, but not always, Fred scaled. So in that case, it might be the likelihood of homozygous for the reference. We saw no reads that supported the reference. We're going to give that a 0. For heterozygous, we'll give it a 0. And for the homozygous for the alternate, we'll give it a 40 because that's a really high Fred score. And we think that that's very much the most likely. But if you have like lower depth or like noisy reads, you could get varying probabilities for each possibility. The one with the most probability is generally the one that's reported in the VCF, but it does give you the likelihood of the others so that you can judge for yourself. In the case of GATK, this is a Bayesian calculation. It takes into account your mapped reads. How many mapped reads did you have that covered that base? What were the quality values of each of those individual bases? And if you have a prior database, the prior probability for that variant. Like, is it a known SNP? If it's a known SNP, then it's like much easier to say that's probably real than if it's not. The quality of calls is also increased by adding more samples.
Is the reference genome service high?
No. So if you were, for example, in the human, doing human sequencing, you can go to NCBI or somewhere else and like dbSNP and download all of the known SNPs for the human genome. And then you can put those in like a particular file format and say, I know there are SNPs at these locations, please go look for them. And I know there's the alternate allele is a C. So if you see a single read with a C, all of a sudden it's much more probable that that's not a sequencing error because you know in the population C's exist at that location. So it's just, that's what the prior is. So you need the reference genome and you need the location of the SNP and the alternate allele. So if you have all of that, that's the full prior. So talking a little bit more about filtering, I talked about this a little bit. The most common is probably a hard cutoff on depth. How many reads do you need to call a sample confidently? This is gonna depend on your ploidy. For a diploid, greater than 20x is very good. The likelihood that you did not sample the alternate allele is pretty low. 5 to 20x is okay. Anything less than 5x is probably not that great. You may be missing. And so this manifests is that you're missing your heads, right? Your heterozygotes are falling out because you have, by chance, only sampled one variant or the other, and you didn't manage to sample both. So if you only have 3 reads, there's a pretty good chance that you didn't manage to sample both alleles. But if you have 20 reads, like the likelihood of you having correctly sampled both copies of the genome is pretty high. Unless you have PCR problems, right? Like that's where PCR problems come into play, because if you accidentally PCR'd one fragment up, it looks like you have 20, you sampled that region 20 times, but you didn't. You had one fragment that you sampled. Does that make sense? Why PCR causes problems? Okay. But then, so high coverage is normally good until it gets way, way, way too high. If it's out of the range of what you expect your coverage to be evenly distributed at, it can indicate a duplicated region in the genome. So at that point, you might be calling like, there's 2 repetitive elements and one has evolved more than the other one. So the variation is between the 2 repetitive elements, not at the same location inside of the 1 repetitive element. If you don't like GATK, some people don't, FreeBayes, SAM tools, know how you say snivers, SNVer, I don't know. Platypus, Varscan, and Vardic, they're all ones I've seen be very successful and get deployed in papers a lot. Freebase would probably be the one I see the most. If you're specifically using GBS or RADSeq data, fast GBS is pretty new and has become pretty popular. Tassel has long been a mainstay in the plant world. I don't know as much about IGST, but I've seen it. If you do not have a reference genome and you're doing GBS or RAD, Unique and Stacks are the two that are going to be designed. They will take your little stacks of reads and create a fake reference for you to map back to if you don't have a reference genome. So those are the two to look for. And briefly, I just want to mention imputation. If one site has low coverage, but is tightly linked to another site with high coverage, the information can be imputed. So if you're missing SNPs, if you have a bit of a sparse matrix, if your SNPs are closer together than your LD, they will travel as a haplotype. So hopefully all of these words are coming back from your genetics class. This is all about linkage and crossovers. So if your DNA is, if your linkage disequilibrium is say 100,000 bases and you have multiple SNPs in that region, even if you're missing data from some of those SNPs, you can figure out the entire haplotype block and impute that for members of your population. There are a number of different software packages to do that. I think the next slide is actually a little bit better. This is what you're gonna see. This is also very much for where you have a lot of dense coverage of SNPs and you have a lot of pre-existing data sometimes. You can also call haplotypes on your population, but you need to be sequencing tens to hundreds to thousands of individuals to really nail down your haplotypes. So let's say these are the actual reference haplotypes that exist in the population. If you sequence an individual, and let's say you're missing 3 sites, and missing this site, missing this site, but you have this 0, 1, 1, and 1, that's enough information that you could have identified that it's the red haplotype and filled in all those question marks. And then you know that there's a recombination from the parental genotypes, and then you can pick up that the green haplotype is on the other end of the chromosome, and then you can fill in the SNPs. So because these things, variations travel in blocks due to linkage disequilibrium, you can impute missing SNP calls. Does that make sense? Okay. Go back, look up haplotypes, look up linkage disequilibrium, think about it. It'll make sense. The final thing you might want to do if you're calling variants is ask if they're actually doing anything functional. I mean, sometimes all you care about is population structure, but sometimes you're looking for a phenotype. Is this causing something? Is this causing some phenotype? So the first thing you normally ask is the variant inside of a gene or a regulatory region. Does it change an amino acid? Does it create a stop codon? Does it shift the open reading frame? All of those things could change a protein and therefore change your phenotype. And there are 3 software packages that you can use for that. SNPF and Anavar both work pretty well. I've never tried Variant Effect Predictor, but it's out there. We are not going to have time to get to that in this class. I don't know. I think I have from last year, I think I have a tutorial on it. So if anyone's really interested, let me know and I'll send you the lab from last year where we did it. Okay, I'm going to go back to this and talk about it while something in the lab is running. So let's get rid of that and move over to the lab. Okay, and I did post instructions for doing the SSH ACF shortcut. So if you follow, they're on the home page of the course. You don't have to do them right now, but like when you're doing your homework and you just want to take a look, there's instructions for how to set this up so that all you have to do is type SSH ACF and it'll automatically fill in the rest of that command. Okay, so the ACF has sent me yet another Q sub command for us all to try. So if you go to, I don't know if I put it in the practical. You have to, yeah, we'll have to copy it from the homepage of the course. I tested it, it did actually work for me, but whether or not it will work for all of us is still. So if this actually works, we can all create a little tiny script in our home directories that will allow us to run this without having to copy and paste it and remember it every time, which is the power scripts. The new part is the :ppn equals 1. I'm not sure what it means, but I think it means that we can all share a computer. All right, that worked for me. Anybody it didn't work for? Okay. It's like, that was gonna be amazing. They all like launched instantaneously. I've been trying to figure out, I must be the only class that we try to teach the interactive terminal where you type each line instead of writing scripts and Q-subbing the scripts, because they've clearly never dealt with this with another class, which I thought was interesting. I don't know. Okay, so for people who did not finish the lab last time or weren't able to get all the way to the end, you can copy the files from my home directory just so you have something to work with. So I have E. coli fixed all the way up through the mapping stage. Oh, the one thing I hate about the Q-sub is the up arrow no longer works, which is terrible. With the SAM and BAM file. So if you need these files, you would get into your home directory, inside the course directory, not in your home folder. Like, it should look like this, but then it should have your username instead of my username. So once you're in that directory, you copy -r .mstate1 fixed here, and then you would run that. I'm not gonna run it because I already have that directory. And let me know if you get permission errors or if that doesn't work. If you finish the lesson on Monday and you're good, you can also, like, let's say you want to save what you did on Monday. You could move your E. coli fix to E. coli fix 2 and then copy my version and name it E. coli fixed Meg's version. But if you do not have this dr fixmate lane sorted.bam, that's where we're starting from. We'll need that. Okay, anybody still waiting for a Q-sub or still waiting for a copy? Waiting for copy. It takes a minute. It's, yeah. We'll see this later in the lesson, but the command to figure out how much space you're taking up is du.skh. I'm up to 7.4 gigs, so it's a lot of, I mean, that stands for disk usage something something human. I have it memorized. We could look at the command pages. So human readable. Anybody else still waiting? One more seconds. Okay, so as you copy this file, everybody, whether you copied or not, this should be your structure. You should be in Lester Haven Courses, EPP622, your username, E. coli fixed, you'll have raw data analysis, FastQC, Thermomatic, and DWA. If you don't have this, copying and pasting the commands isn't going to work. And you'll be typing a lot. And that is on the web page, so you can revisit that. Everybody's copy is finished? Did it work?
I was just checking to make sure.
Okay. You looked very concerned, like it wasn't working. Okay. Okay, so I'm an E. coli fixed. I'm going to go into analysis, and then I'm going to go into 3BWA. So briefly, let me go through symbolic linking. This is a Linux technique. It basically means you don't need to copy files all over the place. You don't want to consistently type like the relative file path or the full file path, because it just takes forever and it's long and it's frustrating. So you just want to be able to use the name of the file wherever you are. And one of the ways to do this is symbolic linking. Why is copying the file to your current location bad? First and foremost, if you have multiple versions of the same file floating around that are all named identically, they're going to get out of sync. You're going to edit one and not the other, and you're going to have a bunch of versions of files, and you're not going to know what any of them contain, and it's going to make no sense. So that's bad. You always want one source file. So any edits to that file there's no other versions floating around. Also, if you change any steps in your procedure, you have to figure out all the places you might have copied that file, and it can be pretty dangerous. You don't know where the file is or what version you're using, so that's not good. So a symbolic link basically allows you to make a file pointer. So it looks just like the file has been copied into your current directory. If you do an ls, you see the name of the file, but it's not actually there. It's basically just this little linker that says the file lives over there. But you don't see that. Well, I'll show you. You can see it if you do an ls-l. But if you just do an ls, it just is a pointer. So if you change the original, that pointer, it's just pointing at the original. You don't have to worry about editing the pointer. So this is an example of what we're going to do. So inside of our 3BWA, we're going to create this little symbolic link, and it's just it's a fake file. It's just a fake little linker, and it's just pointing to this file. But we can say ref genome. So while I'm in this 3BWA folder, I can type ref genome.pasta, and it will treat it as though that file lives in this folder. So I don't have to type the full path. Okay.
If you edit that, does it change? It won't change your original file.
Yes, if you edit this, it changes the original. If you edit this, that linker still refers to this. It's all pointing at the same, it's the same piece of data storage on your hard drive. The whole file is not getting copied anywhere. It's just a little, it's like having a shortcut in your browser where you click on it and it takes you to that site without you having to type in the URL. It's just having like a little button almost, except it's a name. Okay. So we should all be in 3BWA. Let's do ln-s. That is the command for symbolic link. Raw data. And I'm going to start by linking our reference genome. If you want the link to be named something other than the reference genome, you can put a new name at the end, but I want it to just have the same name. So I'm going to hit enter. And now, if I do an ls, I see this nice little reference genome. It's in blue because my terminal is trying to tell me that it is a linker, a symbolic link and not a real file. And if you do an ls-l, you can see it prints the name of the file, the link, and then it gives you this little arrow and basically says, I point to the real file that lives here. So if you ever have a symbolic link and you're trying to figure out where is it pointing, you do ls-l and it will tell you where it's pointing. Yes?
If you copy like the whole folder that it's living in, but edit the reference for that. So like you move it down another folder?
No, the link will not update. The link will, so if I moved this file, the link will turn red and become non-functional. It'll still be in there and you'll just get errors. It'll just say, I'm pointing at nothing. I'm pointing at an empty hole. So yeah, so if you move this file at any point, all your links will break. Yeah. Yep.
I'm sorry, can you do that part again? Because I was in the wrong.
Sure. So
I missed some of that.
Okay. So if I want to symbolic link, let me remove it. So I can remove it, and the link is gone, but not the original file. So we're going to recreate the link. Okay, so that's the whole command, and that's basically saying, in this directory, make a link to this file and name it the same thing that that file is already named. If you wanted to name it something else, you could put another thing here, but I want it to be named the same. So I'm not gonna put anything else on the command line. And now when I do an ls, I can see that file in the directory. It exists, but it's blue, which is, I don't know how many of you have, like, your terminals are giving you colors, but it should probably turn this a different color. And if you do ls-l, it tells you, here's your little blue linker file. It is not a real file, and you know because it has an arrow, which means I'm pointing to the real location, which is this. This is where the file actually lives. Yeah, maybe, sort of. So hopefully everybody's seeing all of these files. Okay, so I want to show you how to visualize alignments with SAM tools, which is really fun. We're going to use that full path to the SAM tools because the ACF version is so out of date. So the first thing we're going to do is index our BAM file. Every piece of software seems to have its own indexing. So BLAST wants to index a database. BWA wants to index a database. SAM tools wants to index a database. Everything wants to index a database, which is fine, except they're all different. So if you index a file with BLAST and then you try to use VCF, it's going to say, no, 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 I want my own index. So like you will end up having like a reference genome indexed by like 6 different programs before your analysis is over. So it just turns out they all have their own little tweaks to how they want to do the indexing. So you just end up rerunning it. Each software package has its own fun index method. But now if you do an ls, you should see this new file .bai. And SAM tools is going to look for that file to exist in the same directory as the BAM file when it looks for the index. It's just like BLAST in that way. Okay, and now we're going to launch the visualization. SAM tools tview. So the first command was index, the second one is tview. We have to give it our BAM file. And then you also want to give it your reference genome. And in this case, we don't have to specify ///raw read /GCF because our reference genome has this little symbolic link. So if I hit G tab, it's going to complete it because it sees that symbolic link in this folder. Has everyone typed that in or has found it on the course webpage? It doesn't on mine, which is driving me nuts. I don't know. What are you using terminal or iTerm? I don't know why mine isn't working. Yeah. Yeah, see, I'm hitting the up arrow and it's just bouncing. I don't know. Okay, I'm gonna hit enter. Nerula, will you help her? Yeah. So it's gonna take over your whole screen. This is kind of like Nano. But this is great. Like, this is so fun. These are your actual read alignments. So your reference genome is being printed across the very top of the screen. And everywhere there's a dot, this is a read with a base that matches the reference. And everywhere there's, so it's a dot if the read is going in the forward direction, and it's a comma if the read is going in the reverse direction. And if there's a base that doesn't match, like this A, that basically means I'm in a column where there's a G, but the read had an A. So you can see in this very global way where your differences are between your reference and your reads. I think those are, I can't remember. Maybe it has to do with quality. Do some people have colors? Okay, so it might have to do with colors. So if you hit the question mark, it's going to give you this little menu, which is all the stuff you can do. The commas are reversed and the dots are forward.
No,
That has.
Underline if that's really is an underline. Okay.
So let's try the colors. I think, yeah, mine, the colors doesn't work either. Does it work for some people? The colors are really nice.
So mine doesn't have
So do your colors match up with where my underlines are? I'm guessing that I'm getting underlines because and colors. I've never seen the underlines before.
Yes.
So you can, I usually have colors. So you can play with like coloring the mapping quality, coloring the nucleotides, coloring the base quality, but I've never seen underline. Oh, you can toggle on off dot view. So if you press the period, it shows you the actual bases. So it looks more like a read alignment. But if you do this, it's really hard to find where the bases are different from the reference because it's just like a big mess of letters. So if you hit the period again, it goes back to the period.
This might be a silly question. Is there a difference between the colors and the
Yes, I think that is, oh, I quit, sorry. And see, mine does this weird thing too where it overwrites itself. You can fix it by typing resize. Okay, we're back. I think the lowercase is lower quality. Oh. So you can hit the, oh, my color started working. I think this acts weird because like I'm logged into a login node and then I'm logged into another compute node and not all the data gets transferred through the tunnel correctly. If you do this on your own computer, it works great. But I still have underlines, which I don't know. So let's go to maybe mapping quality. Yeah, I don't think I have colors. So if you hit the space bar, it goes forward a whole screen. Everything is underlined. I don't think the underlines mean anything. I think it's drawing things weird on our terminals. Okay, so you can go to a specific location. 8690. So I picked this location so that we could look at what will hopefully be called as a variant in this lesson, right? Like, so if you hit G, and then you have to type the equal sign, and then 8690. And escape will make that box go away. So this is a location where we have many, 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 many reads confirming that we have a G, not an A, in the strain. So whatever we sequenced that I randomly pulled from NCBI has a G at this location instead of an A, and we have tons of evidence that that's the case. So this is a good example for what we're going to try to find software to comb through the whole genome and find these for us and report them to us.
Can you print out a section of the, I assume the whole genome scanned through the whole component.
So this is so at the very top we are looking at from base pair 8,671 to 8,700 and about 45. So those numbers at the top tell you what part of the genome you're looking at. So reference genome up here, individual reads down here.
How do you
So this, we only have one sequence because it's E. coli. So if you did have a chromosome, you can type, so you type the G, and you type equals, and then the chromosome number. Let me see if I can pull it up. So there's a whole like tutorial online that you can go through. You type the chromosome number and then you type the location and it pops you there. But I can't remember if between the chromosome and the location. I think there's a colon, but I can't remember. I have to look it up every time. Okay, so this is the manual page for SAM tools, which is all the things that it can do. And I'm gonna look for T-view. Oh, no, that's not what I wanted to do. Okay, so this is how you would specify chromosome 10 colon 10 million. But you don't have to specify the chromosome if you only have one sequence in your reference. You can just hit it, you can just use equals. If you go here and read this, it gives you, I mean, not a ton of information, but a little bit. All right, and to get out of this, you hit quit, Q for quit. All right, questions about that? Does it make sense? There is a homework question that asks you to find a variant and take a screenshot of that variant in SAMTools TView. So you will need to be able to open SAMTools TView with this file, go to a location and take a screenshot. So if you think you're going to have questions about that, now's a good time to ask them. Or to stay after class and practice. Okay. Okay, so the next part of the lesson, I put a whole bunch of links to GATK that were also mentioned in the lecture, so go check those out. So to continue practicing a little bit with the command line, I'm gonna back up and, okay, so if your terminal's doing weird things, type resize, and it should start behaving a little better. So I'm gonna move 3BWA and say 3BWA and GATK. So for me, this is just going to remind me that I'm going to do some manipulations of my BWA alignments with GATK. So by labeling it at this level, I'll know when I go back that that's the folder in which we're going to do the GATK work. I'm going to CD back into that. And you will notice that my symbolic link still works. And if I do ls-l, it still points to the correct location. This still works because it's a relative path. My relative path didn't change. You still go back up 2 directories and down into raw reads. Even though I renamed the folder, that did not impact the location of the symbolic link. Okay, the next thing we're gonna do is create an index file for the reference genome, which we already have one. I think we have to do it again, yeah. We have to do it with Picard instead of with SAMTools. So I'm gonna copy this, we're gonna use, it's a Java program, so you have to start with the command Java. And then I'm gonna put the full location to the jar file because ACF didn't have this, so I just stuck it in our course directory. I'm gonna use the command create sequence dictionary. The flags for this program don't look like normal flags. Again, you just have to read the documentation. So to specify the parameters, you say R equals instead of -R. Picard tools, this is just how it works, just a little different. So R equals, and I give it the reference, and then it wants the name of an output file to store the index. So I gave it the same name, but .dict instead of .fna. And you can name this anything, but it's convenient to name it the same thing, but .dictionary instead of .fna, so that you know that that's an index for the .fna file. And that should go pretty fast. I'll scroll back up. So that's the command. It should give you all kinds of little output. again, it's just creating a Picard style index so that it can be really fast lookup of your reference genome. Okay, now we're gonna use GATK for the first time. It does that indel realignment. So it goes back in and it finds where all those insertions and deletions caused your reads to map all funny, and it's going to go in and clean those up. This is also a Java program. It's called Realigner Target Creator. And there's a whole bunch of flags. So this tells it to use 2 gigs of RAM. This tells it where the GATK jar file is. Again, I put it in our first directory so that everyone in the room has access to it. This is saying the program I want to run is called Realigner Target Creator. My reference genome lives in this file. My input, my read alignment input is this BAM file. And I'm gonna store my output in this file. I'm just gonna use the name of the sample dot intervals. So this is a 2-step process. The first thing it does is it combs through your BAM file and it looks for the regions that look like they need help. It's just finding insertions and deletions and saying, this region looks messy, just write it down, we'll get back to it. And then in the next step, we'll actually go clean it up. So that's what this intervals file is. Basically, it's just gonna be a list of all of the places that have insertions and deletions that need help, that need to be cleaned up. Now, while this is running, you may wonder, like, this is great, I just get to copy and paste it. But when you have to do this, you have to read the manual and you have to write down the command and then you have to fill out all the little flags yourself. And I've done all that for the lab so that they run. But the homework is gonna be more digging into reading the manual and writing your own commands. But these will be nice examples. But what I normally do is open a file with Nano, type the command, write down all the little flags I need, and then go and fill in each little part of each parameter for each flag, put all my little slashes at the end, and then try to run it. But it is a very interactive, like, you don't just start typing on the command line and magic happens. It's very much like you have to go construct all the little bits of code and all the little flags and all the little parameters, get them all in the right order, get them where they need to be, and you're like looking at the document, looking at the terminal, looking at the document, looking. So you're going back and forth, and that's how you build these. It's never as simple as just copy paste. Even though the labs are making you feel that way, when you have to do your own data, it takes a lot longer.
Mine's got like a line of red error lines after the info portion.
What does it say?
User errors occurred, version, etc. For more arguments, your commander incorrect. Gives about.
Do you think you can take a look? Okay. So I would guess if you copied and pasted, it's not likely to be the realigner target creator. You need to make sure this reference genome, the symbolic link, is in the directory you are currently in. You need to make sure this input file is in the directory you're in. And the name of the output file shouldn't actually matter that much. So the 2 things I would start by checking is that these 2 files both exist in the directory you're in, the -r and -i, which is the reference genome and the BAM file. Maybe at the very bottom, it's gonna tell you which parameter it didn't like. But I try to, so if it tells me it doesn't like something about the command, I try to go through in my mind, what's the most likely thing that I mistyped, or what's the most likely thing I messed up, and work backward from there. Some programs give you better errors, and some programs give you very cryptic errors that you can't figure out. So sometimes there's a lot of trial and error. Okay, and now we're gonna do the actual, we're gonna give it back that list of intervals and say, go through, look at these intervals, clean them up, make my insertions and deletions beautiful and pretty and able to be called. So in this case, we're gonna use the same jar file, which is basically just a pile of Java code. We're gonna do specifically called the indel realigner function. Give it the reference again, give it the BAM file again. This time we'll give it the target intervals that we just generated. So we're gonna feed that back in and then store the whole prettified, indel sorted output into a new BAM file I'm going to call alignments, fixed mate, lane sorted, realigned. So I know all of those things have been done to this file. And hopefully that runs for everyone. I tested it last night and it took about 11 minutes. So we're just going to wait on it, and I'm going to go back to talking about VCF files in the meantime. Before we talk about VCF files, does anybody want to ask questions about? Yes. Yes. So yeah, no problem. Here, I'll drag it over from here. Okay, so specifically for packages written in Java, instead of calling the name of the package like we've done with everything else, where it's like samtools or BWA, you have to specify that you want to launch Java to interpret it. And this really gets into understanding the underlying nature of different programming languages, which is outside the scope of what we really care about. The short answer is, when you have a Java jar file, you have to type Java. You just have to say launch Java. That's the only thing that's going to know how to read this Java style file. So you type Java. Java will take some information about how much RAM it's allowed to use. You can do lots of things to specify how much of your computing resources you're going to allocate to it. This in particular is telling Java that it can have up to 4 gigs of RAM. The first command, it only had 2 gigs. The second command, I gave it 4 gigs. You may have to increase that RAM depending on how intense your program is and how much RAM you have available. But Java is just kind of a pain in the butt because a lot of software can figure that out, and it'll just use whatever your computer has available. But Java, you have to just tell it. Usually I go with whatever the software documentation advises. They usually tell you what they think you should put there. You can always just leave it off. Then the -jar is just specifying where is that jar file. You download it from the internet, from like the GATK website, and you stick it somewhere, and then you just have to tell Java where it lives. The -t, so this is, read the documentation, they're all totally different. It's very frustrating. Like BWA, you type, the next word you type specifies what part of BWA to use, so it's mem for us. For samtools, we type tview or index or whatever. For the genome analysis toolkit, you have to use the flag -t, and then you specify exactly what function you want. And it probably has 50 functions. We're only going to see, I think these might be the only 2 we run in the whole class. There's a ton of functions. So we're just saying we want to, for this call specifically, we want the indel realigner function. The -r is for the reference genome. The -i is for the alignments, or the input is what that -i stands for. For the target intervals, so if we come up here, when we ran this command, our output was an interval file. What we want to do is give this back into GATK, so we're going to specify it as the target interval file for the next command. And then finally, we have a new output file that's a new BAM file. A lot of this is reading the manual. Like after that -t, all of the other flags are completely dependent on which function you picked. So most packages are somewhat consistent. Like if it needs a reference genome, it'll try to use -r for every function, but there's no guarantee. You got to go look it up. And it really helps. And what I'm following for this lesson is that workflow. If you can find tutorials, or nature methods papers are great for this. There's like all these tutorials online that'll at least get you guided in the right direction. This HTS-lib mapping to variant workflow. And it looks, I structure my lessons like this because I find this to be super helpful. When other people post theirs, it makes me, like, I'm like, oh, thank goodness somebody showed me what they did and what order they did it in. Because the methods of your papers you read are never this detailed. I mean, it's absolutely, I mean, it's you know, you're just making it up as you go along, trying to figure out someone's methods. So this is the one I picked for this class for us to follow. And you can see, it's like just doing the exact same thing. And I'm just following it step by step. I'll usually go try to read the documentation and see what it's doing. That's a good idea. But if you can find a tutorial somewhere that helps you step through. But once you've run GATK a few times, then it becomes much more feasible for you to read the documentation, read the papers, and start using new combinations of functions. And maybe you say, oh, GATK has structural variants now. I'm going to go use those functions and see how it works and see what I think of it. So if you learn these basics, like how to just run it on the command line, you'll be able to go explore all those other functions. That's the goal. Does someone raise their hand? Okay. Does that help kind of explain the philosophy of what we're doing and how this works and GATK in general? Okay, I am going to talk briefly about BCF files, which is what we will be generating in just a minute. And if anybody else is still stuck, raise your hand or put a red sticky note up and Neurolo will come help you. Okay, SAM tools, BCF tools, and this other piece of software called HTS-lib are all part of a suite of programs. Originally, SAM tools and BCF tools were two different authors, two different groups, and they were doing their own thing. And then they got a grant and they had this big initiative to write this super fast, efficient C library, C is a programming language, called HTS-lib, and to have both of these software tools live on top of that library and both use the library. And it just made everything so much more efficient and unified. So all of these pieces of software can be found in one location, and it's htslib.org. If you go to the old SAM tools page or the old BCF tools page, at the very top, there's a little yellow box that says, for the newest versions, go to htslib.org. It'll get you to the right place. So these three tools. SAM tools is for reading, writing, editing, indexing, viewing, doing anything you might want to do to a SAM, BAM, or CRAM formatted file. We've already seen some of that. BCF tools is kind of the sister program for reading and writing your BCF and BCF files, which we're going to see are variant call format files. And it also calls, filters, and summarizes SNPs. So GATK probably almost certainly does a better job of calling SNPs, but BCF tools is fast and easier to run, so that's what we're going to do. And it's a pretty good first pass on your data. HTS-lib is the C library that we're not going to have to worry about because it's kind of outside of the scope of what we want to do. And here's another link to that workflow I showed you. Here's a list of all the things that SAM tools can view. I mean, we've already seen this with BWA, with BWA, with SAM tools, with GATK, with BCF tools. The primary program that you call, it has lots of different functions. It can do tons of things. It's like a multi-tool. So you have to tell it which one of the functions you want it to do. SAM tools is a little confusing because if you type SAM tools view, I think of that as like looking at something. But what view actually does is convert between formats. And I think it was originally named view because you can take a BAM file, and if you run view, it gives you the SAM file. So it takes it from binary to human readable so that you can view it. But it turns out you use it to go in any direction. BAM to SAM, SAM to BAM, filter. So it does all sorts of things with just converting formats. T-view, we just saw at the beginning of the lesson, which is that text alignment viewer. Super lightweight, quick and dirty. Go look at a location in your genome and how the reads aligned. We're going to see mPileUp, which generates a special formatted file for calling variants. We'll talk about that more when we get there. There's a sort tool to sort the alignments. Merge, so if you do have lots and lots of different samples, different individuals, you can merge all of your BAM files into one giant BAM file, so then you can call variants on the whole thing. Indexing, of course, we've already talked about every program has its own indexer. SAM tools is not the least of those. IDX stats, you can get stats about your BAM file. You can index a FASTA file, which is different than indexing a BAM file, so two different types of indexing, and a whole bunch more. So, but the general format, and if you go to the documentation, it's SAM tools, your subcommand, so view or tview or mPileUp or whatever, your flags, more flags. Some flags need parameters, some flags don't need parameters, so you have to look it up. So you can have a flag and then immediately another flag, or you can have a flag and then maybe that flag needs a parameter, and then yet another flag. So you just have to look at the documentation. So mPileUp format, it is a very verbose format. For each and every base in the reference genome, like I have a single A that starts its chromosome. For that A, it prints its own line, the reference base, the number of reads covering that site, the actual bases of all of the reads covering that site, the base qualities for all of the reads covering that site, and all the alignment mapping qualities for the reads mapped to that site. So super verbose, you will rarely ever use this format. And what we're gonna do in this lesson, and what's pretty normal, is you generate it and pipe it straight to the SNP caller. So way back when we were doing command line, we learned about pipes. We can just pipe the output straight to another program. And that's what you usually do with in-pilot format, is you're taking your band file, you're putting it into a format that is easier to call SNPs on and just shoving it straight into the SNP caller. So we'll see that. BCF tools is a SNP caller that we're actually going to use, but it is also the primary utility that you use to manipulate BCF files. BCF files are variant call format files. BCF files are binary call format. So we learned SAM and BAM. This is BCF and BCF. Yes, more formats. So similar and yet different, of course. So the BCF, the variant call format, has an official spec that you can go look at. And if you're confused by anything in your BCF file, that's the place that you go try to find what's going on. In this case, the header lines start with pound signs, or as somebody in one of my last classes told me, I had to call it hashtags. Hashtag or reference, yeah, no. So pound signs, but hashtags if that makes you happy. Lines with variants are afterward. Now, I think the SAM tools that started with @ signs for the header, this one starts with pound signs. Why is that? Just because that's the way the authors originally wrote it. But each variant gets its own line, and they're separated: header, actual variants. So we're going to go straight to the variants. The header has like the typical header stuff. This is what program ran it. This is what version of BCF is. So the header is quite similar. It's just going to have a lot of metadata about what was done to generate this file. I'm going to go ahead and dig into the variants at the bottom. So this is tab delimited fields. Again, the first field is the chromosome. For us, we're using a single reference sequence, so it will all be the same. But if you had multiple chromosomes, that's where the chromosome would go. The location on the chromosome, so like the base pair, like base pair 3 has a variant, or base pair 87 has a variant. If this is a named variant, like if dbSNP has a record and you have used a prior, that prior SNP ID will go in to the third column. We will not be using priors in this lesson, so you will not see anything in that field. The reference sequence, and in this case, it's not going to print like a whole read or anything. It's just going to say the reference is an A, the alternate is a C. Or the reference is an ACT and the alternate is a deletion. So I'll just give you what the difference between the reference and the reads. The quality score, so this is usually, it's kind of calculated differently by every different variant caller. We'll be seeing MapQ, which is a Phred scaled genotype likelihood based quality value. But that's something that you need to look at whatever is creating your BCF file for how it does quality values. When you filter, you can toggle things from true to false, so whether or not you have passed filter. And then it has lots of information in kind of this like free-for-all field that I'll show you an example of in a minute. And then, like you were asking earlier, Tim, was every column. So if you have 50 samples, there's 50 additional columns. And the genotype of each of your samples is in those columns. So I have individual A has the genotype AA. Genotype B has the, individual B has the genotype AC. Individual C, so each one goes in a different column. And I think I already said this. So BCF is the human readable version. BCF is the binary region, the binary version of the BCF file. Okay, so imagine these. I have them splayed out and labeled, but imagine these in an Excel file. This would be column 1, column 2, column 3, column 4, column 5. So all of this would be on the same line, separated by tabs. The first thing is your reference sequence. So it might be, if your chromosomes are just numbered, it might be 20. For us, we have like NRR something, 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 and that'll be what our reference is. The position, if there's a known SNP ID, if you provided a prior, our reference is a G, our alternate is an A. My map quality value, if this is Phred scaled, it'll vary from 0 to 40 or 0 to 60 or something like that, and lower will be bad and higher will be better. Whether or not it's passing the filter, those are the easy ones. So
for passing and take?
So you get to set that yourself. So when you run a filter script on a VCF file, it'll just flip the filters to false anywhere it doesn't, but you get to set the parameters, right? So originally coming straight out of your variant calling software, they're all gonna be passes, and then you're gonna convert, you know, you're gonna run some filter and say, I only want you to keep sites that are minimum read depth of 10, and everything with minimum read depth of less than that will get flipped to a fail. This is that crazy info field. This looks really similar to SAM tools where there's all these 2-letter codes and I have to go look them up in the specification every time. In this case, NS is a number of samples with data. DP is always depth across the samples. AF is allele frequency for the alternate allele. DB means there's a DB SNP membership. H2 means there's a HapMap2 membership. So there's all these little 2-letter codes that mean a variety of different crazy things. And then you get this format field, and this is where it gets a little complicated. So the format field is telling you for every subsequent column, they're all going to be individuals, and this is the format of the data that those columns are going to be in. So in this case, it's saying for each individual, I'm going to tell you, let's see if I have it, the genotype, print a colon, print the quality, print the depth, and then print something else, and we'll get there. But anyway, so the first thing it's going to tell you is the genotype. The genotypes are usually coded 1 or 0 for reference or alternate. So in this case, individual NA0001 has the genotype 00, so that would be reference. Individual NA00002 is a heterozygote, so we have 1 alternate and 1 reference allele. Individual NA0003 is homozygous for the alternate, so it has a 1-1. The slash versus the bar has to do with whether or not you've done haplotype phasing. If you do not care about haplotypes, it does not matter if there's a slash or a bar here. If you care about haplotypes, then you do care if there's a slash or a bar there. Yeah. The alternate base. Yeah, so if it's like a G, but I've detected in my sample that I also have A's at that position, it's the alternate variant. The reference only says G. So walking in, we only knew the reference. But now we have this alternate, we have this variation that we wanna describe this variation. So it's like, what did the reference have, and then what was the variation we've identified? Whether it's an insertion or a deletion or polymorphism, that's the alternate, it's the variant. Yeah, it's per individual location, like individual location, yeah. The next thing is genotype quality. So in this case, it's Fred scaled in this example, and these are all very high quality, 48, 48, 43. If it was a 0 or a 2, you would think this was very low quality. The read depth is 1, 8, and 5. So this individual only had 1 read covering the space. This individual had 8 reads covering the space. And the haplotype quality. So this is only for phased loci. Again, there's probably a scant handful of people in here who are actually gonna be doing like phasing and haplotype discovery. I can send you more references on that. If you are not one of those individuals, you do not care about this. But this is good information about telling you how well the software thinks it's predicting the haplotypes. And that's why these two have bars and haplotype qualities. This one has a slash, which means we don't know anything about the haplotype. We haven't been able to compute it. So it's probably like humans and cows. I don't know anybody else who would have haplotypes that they really care about. Maybe a few more do. But you have to, okay, so this is not how the file looks. The file is actually a tab delimited, like all of this is on one line, including this format field. That is a field that describes the structure of the rest of the columns. And this will vary from variant caller to variant caller. I know these formats are really complicated, but I think it's good to be able to look at your data. I absolutely, like, it breaks my heart, and I just cannot accept when a student, I'm on their committee, and they're like, oh, well, it produced this file, it's fine. And I'm like, no. That's not how we do science. That's not how science works. If you don't understand it and you can't look at your data, you are failing. Do not do that. I'm on like a quarter of y'all's committees, so you will be hearing from me if you try to do this. So it's complicated, and sometimes you have to like really read the specifications and think about it and look at it, and it's a pain in the butt, and they're very complicated, but they're holding your data. you care about these. As much as you don't think you do, you do. So that's a VCF example. BCF Tools is the software that we use to work with VCF files. It actually does snip and indel calling. It can merge VCF files. The other cool thing it does that I think is really nifty is if you have a reference sequence and you have a list of variants and you want to generate the reference sequence for your individual, it'll go in and swap out all the variants. So now you have a new reference genome for your individual that is customized and has all of the variants that you've discovered. So that's pretty nifty. It has a lot of filtering. It can do statistics. And it can convert between formats. So all of those things are handy. So overview, SAM tools, SAM and BAM files, alignment data, BCF tools, VCF and BCF files, variant data. Yes. You have to download SAM tools as like a program. I mean, I downloaded we have a single copy that the whole class can use. But if you, and ACF has a copy of SAM tools. It's just like more than a year old. The module load SAM tools. It'll give you SAM tools 1.6. We're using SAM tools 1.9 because I think they've done some important updates. But if you want them to update it or you want to have the module load option, just email the help desk and say, hey, I'm a researcher at UT. I need this software. Please install it. And generally they will. It may take them a few weeks though. So it depends on how impatient you are. And it depends on how well prepared you are. So in general, I'm preparing these things like 24 to 48 hours before class and they're not going to get to it in time. I should admit how much of a procrastinator I am, but it is true. But if you give them a couple weeks, they'll generally try to work with you on getting your software installed. And then you would just type module load SAM tools and be able to use it. Any other questions? Oops, take that back. Okay. Did people, did things run? Did people get to the done stage? Yes. Oh, that's so exciting. What is the error? If they copy paste, it works, but if they try to type it, it doesn't. Either.
I copy pasted it.
And it says it can't see the.
It's a pretty long.
Yeah.
Okay. How many people have that error? Okay. If you can stay after for a few minutes, we'll try to sit down and solve it. I may have to Google it. We'll see. I'll try to take a look. So there's a bunch more steps that you can do with GATK. We're not going to do them because they take forever. They each take like 10 minutes to run. We don't want to wait that long. Now you've seen enough of GATK that you can read the documentation and sort out how to do it yourself, right? Yes. So the last little bit of thing, let's index our new BAM file. And I don't think that takes very long. And then I wanted to go through a little bit of cleanup because at this point we're probably eating up a lot of space. Yeah, like hundreds of gigs if you add together all of our space. And this turns out to be a problem, especially if you do want to use the ACF for your research. They charge you for space. So if you start running out of space, that's a problem. So I already showed this a little bit, but let's go back one. du.skh. I'm using 4.4 gigs for just this one folder. That's a lot. And if we go back into it, I do not need all of these files. The SAM file and all of these BAM files are actually easier to recreate by just rerunning those commands than it's just going to cost me money to store them. So you can delete, I would delete the SAM file, and I would delete all these like kind of intermediate BAM files. I would not save anything. And I put a list of what I deleted. Let's see, I'm gonna delete. If you don't want to delete it, you don't have to. If you're like, no, I might have to rerun all of this, you don't have to. But if you're feeling confident, if you accidentally delete the wrong thing, it's gone forever. So be careful. I'm gonna delete that BAM file. Fixmate sorted.bam. Okay, so I've deleted some files. Oh, and I have this like kind of lonely index file that I don't need. Okay, but I've left my realigned files. So now if I back up and type du.skh, I'm down to 610 megs. So just deleting those SAM and BAM files saved me like 70% of my storage space. And take home messages, like your reference genome is not gonna be big. Your FASTQ files, your BAM files, and especially your SAM files are huge. They're eating up all your space. So don't keep the intermediates if you don't. I mean, you can, but if you're running out of space and you need to delete them, go ahead and get rid of them. SAM in particular is a culprit. SAM is huge. If you compress it down to BAM, it's the same file, just smaller. So good to know, because when you go to your PI and you say, I need another $1,000 for another terabyte of storage space, they're gonna be like, what? No. So this is how you, you save space. So we will save variant calling for Monday. Finishing the homework does require you to get through this, so feel free, if things are working for you and you have moved on, feel free to go ahead and go through this. This in particular takes quite a while, so I recommend like setting it up and letting it run and going and doing something else. I think this took like 15, 20 minutes maybe. And the rest of it doesn't take very long at all. But go ahead and give that a shot, and I'll go through real quick on Monday as well. Questions? All right, people who are having errors, who wants to be the guinea pig? Let's sit down and try to figure out what the error is. Anybody volunteering? Do you have the error?